When we're trying to quantify bacteria for Stream Team, we're generally looking at two types of bacteria, total coliforms and E. coli. Total coliforms is a group of bacteria generally considered to be non-harmful. E. coli, on the other hand, can cause illness when ingested. E. coli's presence can also be an indicator of fecal matter or pathogens, and the examples of sources we might expect in streams or rivers can include wastewater treatment plant effluent, leaking sewer systems, animal agricultural runoff, or urban or suburban runoff that can contain pet waste. Shown here is the equipment that we use to analyze bacteria for a Stream Team. We have sample water, but we also have ultra-pure water that has been sterilized beforehand. We also have a powder reagent, a quanti tray, which we will put the sample in, and then a sealer, which will seal the quanti tray to allow the bacteria to incubate for 24 hours. The first step for bacteria is to pipette 10 milliliters of the river water sample collected by the volunteers into 90 milliliters of the pre-prepared ultra-pure water. This is simply to dilute the sample for the processes that we'll do later so that we can properly quantify the amount of bacteria present within the river water sample itself. After you've pipetted the water, the next step is to add one of these powder reagent packets. It's hard to see in the shot here, but in the upper portion it's perforated so that it can be easily opened. When you open it, you want to hold it away from yourself and use your thumb to push on the top to break that perforation. You can then fold it and make sure that you dump all of the powder into the sample. You can gently tap the top of the bottle to make sure that you shake out any remaining powder. After all of that has been added into the bottle, you can close it, and then the next step is to invert it 29 times. An inversion is to tip it over once and back, and you'll notice that that takes some time, so I've sped it up here to make the editing a little bit easier. So again, inverting it over and back until it's thoroughly mixed through. So the next step, we'll use one of these quanti trays, which are basically just a plastic tray with a paper foil backing. They have all kinds of different cells in the plastic shape, and we're going to be using those to segment the water sample into those individual cells, which is important later for when we're trying to count the tray to determine how much bacteria is present. So there's kind of a trick to this. You have to kind of hold it with one hand to keep it open, just making sure you don't reach your hand into it to keep it sterile. And then you'll add all the water, which you'll notice goes down to the bottom. But once you lay it on its side, as the cells fill up, the water will overflow into the following cells, and then you can use the back of your hand or a flat object to kind of evenly distribute that water. In this example, I used more water than we normally do in the process, so you can see me spill a little bit there. So once you have that water distributed, you're going to set it on this rubber tray, and then we're going to pass it through the sealer. That's to close the tray and make sure that it's sealed off to the outside environment. Again, you can see me smoothing it out one more time, and then putting it into the sealer. The sealer that we use doesn't always take them perfectly, so you'll see me shake my hand there to get it kind of locked in. And then it passes through, and then simply comes out the other end. And the sealer seals all the way around the edges, so that once it's sealed, there's no risk of spilling anything. There we go. So I'll demo that now. You'll see me pull it out and turn it upside down. There you go. No risk of spilling. And then you can kind of see that each tray is pretty much totally filled with water with some bubbles. So now that you've done all the mixing, all the pipetting, and distributed everything, the only thing left to do is make sure that you properly label this so that later we know what sample it was, what date it's from, and what time it was put into the incubator, which is important because we want it to incubate for 24 hours. So clearly label and well-written handwriting. There you go. And then once that's done, all you need to do is add it into the incubator. Here I've included some shots of what you would expect those trays to look like, some zoomed in shots too. You can see that some of them have bubbles. Ideally you want to prevent any air pockets, but it's okay to have those in there. You don't need to worry about meticulously shaking them out. And then at the end there you can see that there is that final tray, the largest cell at the end, is actually simply an overflow tray, so you don't need to worry about filling that tray up. Here's the incubating oven that we use for Stream Team. After you've written the time on the sample, you put it into the oven, and it will incubate for 24 hours. After that time's up, we will take all the trays out and we'll start to count them. Any cells within the tray that have turned yellow indicate the presence of total coliforms. Any trays that have turned a cyan color once they're passed under an ultraviolet light indicate the presence of E. coli. After we have those counts, we'll do some math and use those numbers to determine how much E. coli and total coliforms was present in the original sample.